In 2017, researchers at the Scripps Research Institute identified a class of neurons in the human hippocampus with a single, dedicated function: erasing memories. Not storing them, not retrieving them, erasing them. Every night while you sleep, these cells activate. And neuroscientists still cannot account for how your brain decides which memories never wake up with you. The hippocampus sits 2 inches behind your ear. It is the size of a curved finger. For decades, neuroscientists treated it as the brain's filing system, the place where raw experience gets compressed into something permanent. That was the model. Memory went in, memory stayed in. Forgetting was failure, hardware degrading over time, the natural consequence of an aging system under accumulating stress. The Scripps researchers were not looking for what they found. They were mapping memory circuits in mice, tracing the molecular fingerprints left behind after a new experience. The mice learned a task. The researchers tagged the neurons that fired during learning. Then they waited. What they observed during the sleep cycle that followed was not consolidation, it was demolition. A specific protein, ARC, was flooding certain synapses. ARC does one thing: it dismantles the receptor structures that allow neurons to communicate. Where ARC appears, connections weaken. In sufficient concentration, they vanish. The tagged neurons, the ones that had just encoded a new experience hours earlier, were being selectively flooded with it. The brain was not filing the memory. It was deciding, simultaneously, which parts to keep and which parts to destroy. And it was making that decision while the mice were completely unconscious. Here is the obvious explanation. The brain has limited storage capacity. It cannot hold everything. Sleep is the maintenance window, the overnight cleanup that clears the backlog, so the system runs efficiently the next day. This is the synaptic homeostasis hypothesis, proposed by neuroscientists Giulio Tononi and Chiara Seierle in 2003. Synapses strengthen all day as you accumulate experience. Sleep downscales them back to baseline. You wake up lighter, literally, synaptic load reduced by roughly 20% overnight. The brain prunes the irrelevant to protect the essential. Tidy, logical, almost satisfying, but it doesn't hold. The problem is selectivity. If sleep were a simple bulk reset, a proportional compression of everything acquired during the day, you would forget evenly. A little of everything, roughly equal. That is not what the data shows. Certain memories survive intact. Others, experienced with the same emotional weight and at the same hour, are completely gone by morning. Not faded, gone. And the deciding factor is not intensity, not repetition, not how much attention you paid at the moment of encoding. Researchers cannot reliably predict which memories will be erased and which will persist. There is no consistent rule. Worse, the deletion is not passive. The brain is not simply failing to consolidate certain traces. It is actively targeting them. In research following the Scripps work, a distinct synaptic structure emerged in memory circuits, one with a molecular architecture built specifically to receive and amplify the ARC erasure signal. Not broken receptors, not the result of damage or degradation, a purpose-built feature. The brain engineered infrastructure specifically optimized for receiving the erase command. And here is the detail neuroscientists have not been able to account for. That forgetting infrastructure is not uniformly distributed. It clusters. In certain individuals, in certain memory circuits, the density of erasure machinery is 2 to 3 times higher than in others. You could be standing next to someone, both of you experiencing the exact same event, and your brains will delete different things tonight, because one of you has more forgetting architecture installed in that circuit, not because of how you felt, not because of what you did afterward, because of a structural asymmetry no one chose and no one can predict. The mechanisms proposed to explain the selection process range from metabolic tagging to glial cell signaling to the precise oscillation frequency of your sleep spindles, the tight bursts of neural activity your brain produces during deep NREM sleep, cycling at 12 to 15 times per second. Some researchers believe the spindles are the selection signal. That memories not synchronized to the spindle rhythm simply do not survive. No one has demonstrated how the spindles know which memories to synchronize with. The actual decision, what gets invited and what gets cut, remains outside the model. Researchers can describe the mechanism of erasure with precision. They cannot explain the criteria. Every single night, your brain runs a process it has never fully explained. It reviews what you experienced. It builds some of it into lasting structure. It deliberately destroys the rest. You will never know what it deleted. You have no access to what was removed, no awareness it existed. By the time you open your eyes, the decision is already made, the evidence already gone. The question neuroscientists cannot answer is not what the brain is deleting each night. It's whether the version of you that wakes up is missing something it needed.